Thanks everybody for coming back promptly, and we're going to continue our session. Harvey Clewell will be our next speaker, and Harvey's going to be talking about modeling data from individuals to populations.
I will be focusing particularly on using in vitro data from individuals to predict the variation in vivo in populations, not interpretation of epidemiological studies, for example. And I'm going to have kind of a risk assessment focus in that I believe that the guidelines that have been developed by the IPCS for chemical-specific adjustment factors give us a good way to try to incorporate some of these issues in quantitative risk assessment. And so I want to also clarify that there's always been two parts of risk assessment. There's the quantitative part where you generate a number, and there's the qualitative part where you generate concerns. And so the concerns generally end up as uncertainty factors or safety factors or just concerns, unresolved concerns. But in most risk assessments, there is some amount of quantitative number crunching that results in some value. And I've been trying for years to try to help risk assessors to be able to move more of their concerns into quantitative changes to a particular
We're sorry. Your
guideline.
Conference is ending now. Please hang up.
So, thank you. So that wasn't so bad. I will talk first about the IPCS chemical-specific adjustment factors. The EPA also has this concept of using quantitative alternatives or replacements for the more qualitative uncertainty factors. And then I'll talk about approaches for quantifying first pharmacokinetic variability, because we've actually got a lot more of a track record in that area. And then I'll talk about some initial inroads or some considerations for doing the same thing for the pharmacodynamics. And I'll finish by looking forward a little bit. So the some years back, IPCS, it's part of the WHO, developed the concept of chemical-specific adjustment factors, and there's a guidance document at this website. And it was a very thorough evaluation, multinational, and tried to consider all of the various aspects that one must include in order to come up with a quantitative adjustment for across species and also within human variability. And the idea was to have some generally agreed process for replacing the usual uncertainty factor of 10 for animal to human and 10 for human variability with something that was a bit more chemical specific. And so first of all, the CSAF for toxicokinetic variability is based on a comparison of either a directly measured or modeled surrogate for the internal exposure to an active entity. Andy Rennick was actually the one who wrote up the document, and so it tends to reflect his choice of words. But examples of the kind of things one might compare are the area under the curve in the animal and the human, or in an average individual versus a more sensitive individual, or even a sensitive subpopulation. And in some cases, it may be that Cmax is considered to be a better measure for a particular mode of action, or in some cases the data might be available as clearance, which usually is inversely proportional to the AUC, but not always. And in principle, this data could be derived from in vivo kinetic studies in humans. My working assumption is there's not going to be a whole lot of that going forward with environmental compounds. This certainly is the case of what is done in pharma, but in environmental risk assessment and toxicology, it seems unlikely that controlled human studies are going to continue to be a major part of our data set, with the possible exception of microdosing, which is still quite problematic when you get outside of pharmaceuticals. So that's my assumption. But so what's happened in pharmacokinetics is we're moving away from relying on in vivo controlled dosing to try to predict the in vivo pharmacokinetics based on in vitro data, particularly metabolism data, and modeling. Of course, it's complicated, yes? And so there are a large number of factors in a population that can affect the pharmacokinetics in an individual, some of them genetic, some of them environmental exposure related, age, weight, health status. And the point that I want to make is that if you conduct a Monte Carlo analysis where you draw from reasonable distributions for each of the parameters in a physiologically based pharmacokinetic model, that you can try to estimate what would be the impact of the pharmacokinetic variability across a population. And this just shows a histogram from the ETER website. Mike Darson is among us and we'll be talking later. He actually bought me a beer the year that we did this risk assessment for methylmercury and came up with a distribution of RFDs across the population instead of just a single number. This then considered the variability in kinetics of methylmercury across the population. That's across a population which, in that case, you're worrying about adults by default. But in fact, of course, the population also varies with age. And particularly in early life, the most random process in development is the actual birth event. And so the level of development for a child which is 6 months old really depends to a large extent on how old they were when they were born and how rapidly they're developing compared to their peers. And so particularly looking at early life, it's important to consider population variability. And so once again, physiologically based pharmacokinetic modeling does allow us to look at that. And so what we can do is we have data, particularly in the human, we have a wealth of data on the age-dependent changes in organ weights, in blood flows, in enzyme ontogeny. So we can actually, and also glomerular filtration, all the biological and biochemical processes have been studied very well. And so we can actually incorporate all those into a model where the parameters all change with age. And then we introduce a chemical having characterized which enzymes metabolize it, and then we can, and using the adult levels, then we can ratio to get the early life values that would be corresponding, and then we can model the blood concentration over different ages at the same at the same dose under a given metric. In this case, I'm going to show you a constant dose in terms of milligram per kilogram per day, but you could do a constant milligram per day. You could adjust by actual exposure differences between early life and later, however you want to do that. But the point is that the model factors in the change in the pharmacokinetics of the individual. And this shows two compounds that we looked at. We looked at a number of them, but this provides a nice contrast. In one case, the green line is dioxin, and the red line is nicotine. Nicotine being water-soluble, and TCDD, of course, being highly lipophilic. For nicotine, it actually mimics what you generally see with drugs, which are also water-soluble, that the exposure in early life tends to be greater than the exposure in adults because of the ontogeny of the enzymes responsible for the clearance of the chemical. And this has been characterized by Rennick and others as being on the order of perhaps a factor of 3, roughly, for most compounds. For TCDD, you see a much more complex time course, which is, as you might guess, due to a number of different factors becoming important at different ages. Overall, it mostly goes by fat fraction. As you increase your fat content, you increase your dioxin levels. The dip, this is one of the things they call an emergent property. What that means is you wrote the model, but you don't understand the result that it predicts. So the model predicted that whereas the infant was born at the same concentration of TCDD as the mother, which makes, of course, sense, 6 months later, it's only half as great. All the women in here are thinking 6 months, so you should double your birth weight by 6 months, right, a normal infant. So sure enough, this is just growth dilution. And so that's what's predicted by the model, because the rapid expansion in volume is the main difference in the dioxin concentration internally. And I'm a scientist, so I'm a skeptic, right? So I wanted to actually see something besides that aphorism. And so I was able to find some data from Filser's lab in Germany, where they'd actually measured concentrations in the infants of different ages, and saw the same at the bottom here. The U-shaped curve is for those that are not breastfed. If of course, the infants are breastfed, that introduces a very potent source of dioxin. It's a very different case. Yeah, there is something a little going on with this button thing. So then a second dimension of the variability in the population that we can look at with the PBPK modeling is genetics, differences in phenotype, I would say, in the case of metabolism. And so an important point that I would like to make, because it has a direct analogy to looking at pharmacodynamics, and that's if you look at the difference or the range of values or the ratio of values from a normal population to a sensitive population, you can draw a conclusion that may not actually track with observations across a population, because there are so many factors that are varying at the same time. And so something that seems like it would make a big difference looked at in isolation may not actually make that big a difference on a relative scale with all the other changes that are also true across the population. And so this shows a case for Coumadin, also known in the environmental world as warfarin. So it's a rat poison and it's also a drug. These are various vectors that were used in order to estimate the maximum capacity and the affinity for clearance of Coumadin. And there are 3 different alleles, and there's both homozygous and heterozygous in the population. But so we use this data to estimate the relative metabolism clearance capability for Coumadin, depending on which allele a person had. And we had prevalence data, and there's kind of data is available for most, certainly all the CYPs, but most enzymes. We have this kind of prevalence data, not just in the U.S. population, but in other populations around the world. And you can see that the most prevalent is the S1 homozygous. It also happens to be the fastest clearance. And the least prevalent is the S3 homozygous. It also happens to be the slowest. And so if you run the human model with just the change in the metabolic clearance, you see these are the time course curves for a single dose. And so it's obvious that there's a huge difference in the exposure, the blood exposure to Coumadin for these three subpopulations. And of course, everyone probably knows that Coumadin is one of the drugs that they have to be very careful with in the clinical situation, because they can cause bleeding in some patients. They have to monitor their blood concentration and adjust the dose accordingly to keep it safe. But now if you take this difference and you incorporate in with it all the other things that change, the fat content, the other metabolic pathways, glomerular filtration, all the things that vary across a population, then what you find is, in this case, the dark bars are the total population, and the white bars are the quote normal population. So there's I think I might have that right. So what you see is for the total population, the dark bars, it's a little bit more skewed to the right, and that's because of this polymorphism, but because of the distribution of the prevalence of that polymorphism, it has a fairly small effect on the population. So that's why it's important to look at this kind of translation from an in vitro situation or from a single component situation to a multiple. This is also part of the reason it's so hard to find associations in epidemiological studies, because something that's really important can get washed out by all the other things that are going on. this, of course, is, as always, it seems, in pharmacokinetics, the pharma has done it first. And Jeff Tucker at the University of Sheffield, it's a great success story of someone who saw a problem, drug-drug interactions being a concern, and saw the solution, which was predicting the in vivo kinetics of the two compounds that might have competitive inhibition for a particular isozyme from in vitro data. And this is now accepted by FDA and other international organizations as a way of screening drugs for whether there's a danger of drug-drug interactions between that drug and the other drugs that a person might be on. The development of SimSip, which is the name of the platform, was funded by a number of drug companies, still is being funded, and it supports just this kind of in vitro to in vivo extrapolation like I showed you with Coumadin, but on a grander scale. And I use SimSip now instead of doing it the hard way. And they have data on different populations, including disease populations, on pediatric populations, so you can look at the early life exposures. And there are some limitations for use with environmental chemicals as opposed to drug-like compounds. In particular, the focus for a drug-like compound are the CYPs and the UGTs, the glucuronide conjugation, but not GSTs or esterases, which are very important for environmental compounds. And so this is one of the areas where there needs to be some expansion. And I know for a fact that the folks at SimSip will be glad to try to add that if someone in the environmental world would give them some money. But it never makes the hit list for the drug companies that are funding them. I don't want to make it sound like I'm pitching SimSip. I tend to say nice things about products that I really like, but there are other platforms that also can do this, GastroPlus and PKSim, which are also developed for pharma use. And so I guess in general I would say they might be more powerful than SimSip, but a little harder to use because it's more on the user to provide the information that they use. So this is a well-embedded technology in pharma, accepted by the agencies, that really should be just taken over and applied in the environmental area to characterize variability, the effect of variability in the human metabolism on the internal exposure at the same external exposure. This just shows that you input from recombinant CYP studies what are the rates of metabolism for a compound. These are all done with in vitro studies. They can be done high throughput, and there are the technology for converting those recombinant results to whole cell to primary cell equivalents is in place. And then this shows the kind of output that you can get. This is from a SimSip publication, and it shows that in addition to the prediction for average individual, you can get the total population percentiles. Okay, so I think we're a long way on being able to come up with data-derived or chemical-specific adjustment factors in kinetics. Now, dynamics is clearly a bit tougher, and these are actually quotes from the CSAF from a talk that Betty Meek gave about the IPCS guidelines, and the conclusion was that there's rarely sufficient data to replace this component, and most of the data that you get addresses both kinetics and dynamics, and trying to deconvolute the kinetic contribution to variability is difficult, and so very often you don't have the proper data. And what was suggested was that data could, in principle, be obtained from in vitro studies in the tissue of the critical effect from an average versus a sensitive individual. And that is there's an example of using red cell lysis in the case of 2-deoxyethanol, I think it is. And so it is, in principle, doable. One of the things I'm looking at is with arsenic. I was able to get from Sam Cohen snippets of uroepithelial cell, actually urers, from kidney donors, and then we exposed them to a mixture of arsenite and its metabolites at the same relative concentrations that you see in human urine, but at total arsenic concentrations ranging over 2 orders of magnitude. And you can see here the dose response for heme oxidase, showing that the response of these cells in the oxidative stress pathway is fairly well defined across these various individuals. There's a good deal of variability, but there is a commonality as well. And for the genes that were expressed most uniformly across individuals, what I didn't show here, probably should have, was a Pca analysis that shows that if you look at the response from different individuals with a Pca plot, each individual comes out in a different place, completely isolated from the others, and the dose response for arsenic is actually small effect on the genes compared to the difference in the gene responses from one individual to another. So that's the toughest part of the heterogeneity. But so in order to, what I did see was that the minimum concentration at which you saw any genomic response was roughly similar across the individuals, despite the fact it might not be the same gene in each case. But then what I did was take the genes that were most commonly expressed, and they turned out to be really, they make a lot of sense from an arsenic mode of action viewpoint. Oxidative stress protein folding, metallothionein, thioredoxin. The interesting ones to me, anyway, are the DNA damage sensing and the immune response, because I think those are actually the key ones for a carcinogenic evaluation. But so then on the right, what we did was apply Rusty Thomas's approach of doing benchmark dose analysis on these genes, and we saw a commonality in the range you can see between 0.1 and 0.6 micromolar is where you see all of these genes coming on board. And for any given gene, it's basically a factor of 3 or 4 between the lowest and highest benchmark in an individual. And so this is a way of getting a handle on how much pharmacodynamic variation is there in a population. Certainly the N here isn't large enough to be able to come up with the uncertainty factor you'd want in a risk assessment, but it certainly gives us a beginning of an idea, and I think it's a direction that we can go. And looking forward, I think that for kinetics, we want to follow in pharma's footsteps and collect some additional data that supports non-drug-like compound information. In dynamics, I think that there's a potential for these high throughput in vitro studies with cells from multiple donors. And Yvonne Rusin said that I should also say or cell lines. Like I said, I'm a skeptic. So someday I might be willing to accept the idea of multiple cell lines. I'd be, it could be critically dependent on the extent to which we can characterize whether the cell lines are still representative of primary cells. And what I'm looking into now is trying to work with induced pluripotential stem cells and to see to what extent they might be a way of looking broadly at population variability across chemicals with cells that still are, in some sense, normal. And when we looked at arsenic, we did a literature search of arsenic genomics, and the things that became apparent is that immortalized cell lines seem, in general, to give similar results to primary cells, but tumor-derived cell lines don't. And so you just have to be careful how the cells differ from a normal primary cell. Then what we're doing at the Hamner is then trying to develop biomarkers, particularly high-content analysis biomarkers for specific toxicity pathways, like H2AX for DNA damage detection, or NRF2 for oxidative stress, and NF-kappa B, BCL2. There's some key proteins that we can follow so that you're not up at the genomic level. You can actually be looking at the protein level, including phosphorylation, so that you can actually then measure the concentrations at which a particular pathway response is exerted in different individuals. And I do want to say that I I'm working with Yvonne Rusin on the multiple mouse strain study interpretation. So I think that's a great way to go. This just shows, what I'm doing right now is just trying to see if we can characterize the strain variability. This is trichloroethylene, and then we will compare the apparent variability in the metabolism and, well, in the internal exposure to the different mouse strains with that which we can, in the case of trichloroethylene, characterize very well in the human, because there have been Markov chain Monte Carlo studies done with trichloroethylene that give us a good handle on the variability as opposed to uncertainty in trichloroethylene internal exposures in the population. And actually, Wei Shui did the most recent MCMC analysis for trichloroethylene. And so the posteriors from that analysis can be used to compare with what we determine in the mouse. But so that's I would say that right now the multiple mouse strain technology is not ready for prime time quantitatively to replace an uncertainty factor, but it can go in the right direction. And that's it. Thank you.
Thanks, Henry. Lou Gross, National Institute for Mathematical and Biological Synthesis at the University of Tennessee. So the approach that you talked about was very much assuming that the population is just made up of a bunch of individuals, that it's just a sum. There are other approaches to population modeling, of course. And so I wanted your advice and opinion about whether or not they're potentially more applicable. And so these are approaches in which you actually track individuals, so agent-based models, individual-based models, and so on, that take account of interactions between individuals. So under what circumstances, from your experience, would the variability associated with the interactions that occur in terms of common exposures, in terms of heredity, and so on, that drive population-level responses would be appropriate relative to just the variability in pharmacokinetics and so on within individuals? Do we know yet?
No, we certainly don't. I think that's a really interesting area. It's kind of folding in the exposure part, which I was avoiding, and it's really more population oriented as opposed to starting with individual data and moving to population. So that's the reason I didn't bring it up. But it certainly is when you're actually trying to interpret these things. Those are areas that need to be considered in trying to do a Monte Carlo or Markov chain Monte Carlo simulation of a population. I think that we generally assume independent distributions, and they're certainly not.
Harvey, very nice presentation. Bill Slicker, NCTR. Just a question about moving back from that neonatal period into pregnancy and how some of the models that have been developed and approaches may work when you're looking at conception through birth in terms of modeling and looking for drug-drug interactions, et cetera.
Well, I want to thank you for that softball. Yes, as you know, that is an area where PBPK modeling has been demonstrated for a number of years to be quite useful. We have really good information again on the changes during gestation and the early neonatal period, so that just as I showed with the birth through 70 years, actually when we ran that simulation, we ran an adult model for dioxin in particular. We ran an adult model for some like 30 years, a female adult, and then ran a gestation model for 9 months, and then started that model. And we also, I didn't show you, but we also ran a lactating mother model. So all those things can be done within the framework of PBPK modeling. And actually what we're doing right now with those, I've mentioned to a couple of folks, but Matt Longnecker at NIEHS has come up with an idea he calls reverse causation, where physiological changes associated with different endpoints like early menarche, late menopause. Thank you. I don't have that worry. So, even low birth weight can induce a change in concentration of a chemical that could cause an association between the chemical and the effect when actually it's driven by the effect. And these models are proving to be really valuable for that, for evaluating epidemiological information. He's actually trying to determine whether you can recapitulate an odds ratio that way using the variability again. So I think that age-dependent modeling is probably one of the most informative things that we have to contribute to population variability.
Thank you.